Yeah, thank you very much for this very kind introduction. So I was not very inspired actually when you asked for the fun fact, and it's the only thing I could really find. So thank you very much for the invitation. I'm very glad to be here. So I'm now leading a team which is joined between two labs, one at Sorbonne University in Paris and one at the Institut Curie. I will move soon to Institut Curie in a couple of years. To start, today I will present recent work we developed in the lab dealing with developing tools to control biological processes using chemically induced dimerization. And so, as it's been said in the introduction, oh, yeah, sorry, like this. Yeah, so I'm a chemist by training, so of course, when I see a cell, I don't see really exactly maybe the same thing that you guys, biologists, see. What I see, it's really a very complex chemical system. And for a chemist, really, a cell is really one of the ultimate playground in terms of reactivity, selectivity, and analysis. And really, it's really a strong motivation for the work we do in the lab. And of course, one of the really intriguing and fascinating question for a chemist like me, it's really how such complex chemical system, which is composed of more than 1 billion individual proteins encoded by more than 20,000 protein coding genes. How does this chemical system achieve specificity in their function? And I think you are all well aware that cells achieve this kind of specificity by organizing functionally interacting proteins in space and time through different approaches. So in particular, in terms of spatial organization, some specificity can come from the compartmentalization of functionally interacting proteins, the colocalization of these proteins to membranes in particular, or the organization of protein complex assemblies through interaction with specific scaffolds. And this spatial organization is really key for almost all processes inside cells, going from gene expression regulation to protein and organelle transport and so on. And so it is really in this context that we started to think of developing tools to try to develop tools that allow to control the organization of functionally interacting proteins in space and time. And so now I'm more in the field of chemical biology, and chemical biology has provided in the last decades different tools to try to study the role of spatial organization and protein proximity in different biological processes using a very interesting concept, which is called chemically induced proximity. And the idea of this chemically induced proximity tool is really to, so I don't know if you see my arrow on the, no, yeah, you see it? Yeah.
Yes.
Right. So this concept of chemically induced proximity tool is really simple. It's basically you can fuse to two protein A and B. You are interested in studying the role of their proximity. You fuse them to two dimerization domains here called DD1 and DD2, which only interact in presence of a small molecule that we call a chemical inducer of proximity. And this really allow to have experimental control on the proximity of two proteins, and has been really essential to study different kind of processes, including the control of transcription, signal transduction, chromatin regulation, protein transport, and so on. And so there are already many tools that exist in this field. And here it's a summary which has been done by Gerard Crabtree a few years ago in this science review that describe many different chemically induced dimerization tool. And probably one of the most famous one that you might know, it's the one that uses rapamycin as a chemical inducer of proximity that allow to dimerize two protein domains that are called FRB and FKBP. And so there are many others. I will not really go into the details of these different systems in the interest of time. But just as a summary, there are two categories of systems that use, like in the case of the rapamycin system, small molecules that act as molecular glue that will really just allow, that will bind one of the two protein domain and allow the interaction with the second one. And the second strategy is just to use bivalent molecules that are composed of two parts that bind respectively to the two dimerization domains, allowing to induce the dimerization. This approach is interesting because you can sort of use non-natural molecule to induce dimerization. The problem is when you use bivalent molecules, you can have a possible hook effect, meaning that the concentration of the dimerizer you will use will be important, because if you use too high concentration of dimerizer, you will just interact with the two protein domain and no interaction will occur. So recently, actually, a third strategy has been proposed, and which were very interesting for us, which is actually to take a protein ligand complex and to actually split the protein part in two complementary fragments in such a way that upon addition of the ligand, you would induce the dimerization of the two complementary fragments. So this, in theory, could be a very general approach because you could really imagine to develop chemically induced dimerization tools from any protein ligand complex. It really acts here as a molecular glue, like the first case here, so you have no hook effect. You can imagine to have very small fragments here, depending on the protein you start with. Which can be useful also. And what was also very interesting for us is that you can add additional properties. And the one additional properties we were interested in, it's actually to add fluorescence in the process and to develop a system where basically upon chemically induced dimerization, you would have a fluorescence readout that could be used to really certify that the dimerization process occurred. And to do this, the idea it's very simple. It's to develop chemical inducer of proximity that would become fluorescent upon dimerization of the two dimerization domain. So this was really the starting idea to develop chemically induced dimerization tool with a self-reporting capabilities or with a fluorescent readout. And so to design such system, we actually wanted to apply the strategy I described to take a protein ligand complex and to split the protein in two parts. And so to do this, we started from from fluorescent chemogenetic reporters we previously developed in the lab. So for almost 10 years now, we've developed a collection of fluorescent reporters, which are chemogenetics, which are composed of a protein part, which can be genetically encoded, and a synthetic part that provides the fluorescence. And these chemogenetic reporters can really be an alternative system to classical fluorescent proteins you might use, or also to self-labeling tags you might know, like SnapTag or HaloTag, which are used to covalently label proteins with dyes. So in these systems that we called FAST, which stands for fluorescence activity and absorption shifting tags, these systems are composed of a small protein that we called FAST, which is only 14 kilodaltons, so it's half the size of a GFP. And so you can virtually fuse it to any protein of interest by your favorite genetic tool. And this protein tag can bind and stabilize the fluorescent state of this kind of fluorogenic chromophore. So basically, these chromophores are non-fluorescent when they are free in solution. They are only fluorescent when they are bound to the fast tag. So this allows to really have a high contrast even in presence of the fluorogenic chromophore, and allow to obtain this kind of image just by addition of the small molecule. And so the interesting feature of this kind of technology is that we can really use them in different systems. So we could demonstrate that we can use them for imaging proteins in live cells. It's compatible with super resolution technique like STED, and it's also compatible with imaging in multicellular organisms such as zebrafish larvae, for example. And another interesting feature of this chemogenetic system is that they are composed of two parts, a protein part and a synthetic part. And we can play with the structure of the small synthetic chromophore to actually modulate the spectral properties of the system. So we have a version of FAST that we call PFAST, which binds this hydroxybenzylidene rhodanine chromophore that can be green, yellow, or red. And we even evolved recently a new version that we called near-infrared fast, NIR fast, that binds this time chromophore with elongated pi system, which now emits in the far red and near-infrared range. And interestingly as well, these two tags are actually can be orthogonal depending on the chromophore we use. And so we can do two-color imaging like this image here or this movie here. So this is the fast technology, and the idea was actually to take one of these fast system and try to develop a chemically induced proximity tool simply by splitting the protein part. And the idea was that upon addition of the chromophore, we would expect that we would promote the interaction of the two complementary fragments. And as the chromophore lights up within the protein binding cavity, you would form a fluorescent ternary assembly that would allow us to have the self-reporting capability we were interested in. So to do this, we used our PFAST protein, so this one that binds these chromophores. And we splitted it in two complementary fragments in between the position 114 and 115, giving two fragments of very different size, a very small one of only 11 amino acids and a large one of about 114 amino acids. And interestingly, what we observe is that upon addition of the fluorogenic chromophore we use for PFAST, we actually could really promote at low concentration of fluorogen the complementation of these two systems. So we could really have a system that upon addition of the chromophore would promote the binding of the two protein fragments. And it's like this that we developed, so the technology catch fire, it's been introduced in the introduction. So catch fire stands for chemically activated tethering of chimera by fluorogenic induced recognition. And so in that case, we sort of renamed some part of the system to make it a bit more catchy. And so we renamed the N-terminal part of PFAS, so the large fragment, we called it FireMate, and the C-terminal domain of PFAS, the small fragment, we called it FireTag. And we also renamed the chromophore, the fluorogenic inducer of proximity. We call them match now because they can match together the two proteins and light up at the same time. So this work, it's the postdoctoral work of a very talented postdoc who unfortunately left the lab, Sarah Botone, and it's a work also in very close collaboration with Frank Perez, you might know, with a cell biologist at the Institut Curie in Paris. So how does this catch fire technology work? So here it's a very simple experiment to demonstrate catch fire in action. We have a cytosolic protein, mCherry, which is fused to FireTag, and the FireMate, which is anchored at the surface of mitochondria through a fusion with a TOM20 receptor. In absence of the match molecule, the two proteins do not see each other. So you have already a cytosolic protein here in red and a mitochondrial protein here in blue. And upon addition of match, what you see, it's almost instantaneously relocalization of a subpopulation of the red fluorescent protein at the surface of mitochondria, and also the appearance of a green fluorescence coming from the formation of the ternary assembly. So what I will call now the catch fire channel. So here you could see that it's very quick. And we'll show you other experiments demonstrating the temporal resolution of the approach. So to do this experiments, we use this small molecule that we called match 550. And the 550 subscript, it's actually the wavelength of emission of the complex formed by the assembly. So it emits at 550 nanometer, so more or less in the green-yellow region. But so interestingly, we can actually modulate also the spectral properties of Gautier by actually modulating the chromophore we use. As I told you, for PFAS, we have different chromophores, and depending on their structure, we actually can have different spectral properties. And so we also have a match 600 that emit at 600 nanometer, which have this chemical structure, and that allow to do a red Gautier. So in that case, it's the very same experiment than the one I just showed you, except that we used ECFP fused to EGFP, sorry, a green fluorescent protein fused to fire tag, and we induce red Gautier using this match 600. And for some experiments, maybe you might be interested of not having any fluorescence, even though the fluorescence is the cool part of Gautier. And in that case, we also have a dark match that is non-fluorescent at all, but that can anyway allow to induce Gautier, as you can see here on this experiment, where we could recruit a cytosolic protein at the mitochondria surface, but we have no fluorescence at all in that case green channel. So we could demonstrate that Gautier could really work to recruit cytosolic proteins at the surface of different organelles, at the surface of the Golgi, the endoplasmic reticulum, and also at the surface of the plasma membrane, and that it works very efficiently in all conditions. What's very important for this kind of chemically induced dimerization tool, it's of course that you want to eventually use them to really have a temporal control also of the process. So the temporal control can come from the addition of the small molecule to induce the dimerization, but of course it can also come from the stopping the interaction by simply washing out the small molecule. So ideally what you want is really something where you can switch, induce interaction by addition of the small molecule, and ideally to stop the interaction simply by washing out the small molecule. So the interesting feature of the system that the match molecule we use are highly cell permeant, and we can really induce rapid dimerization. But it's also actually washable, and like this we can really turn off the interaction. So here it will be a movie that will show this actually. So it's M-cherry protein fused to fire tag, and this time the fire mate is localized at the surface of the Golgi through a fusion with giantin. In absence of match, we have no interaction of the two proteins, and if we add match, we will see a recruitment of the red fluorescent protein at the Golgi. But if we wash away the small molecule, you see the disappearance of this signal, and and you can add again match and wash it away again. So this movie just show you really that the dimerization process could be fully reversible simply by washing away the small molecule. And of course, with this movie, we could really quantify the kinetics of the association process and dissociation process. And we could demonstrate that basically the half-time of the association is about 25, 30 seconds. And for the dissociation, it's more or less the same. So it's basically simply the diffusion of the small molecule across the cell, and the cell uptake, which control the temporal resolution here of the association and dissociation. So then we wanted to demonstrate different uses of this approach. And the first thing we decided to study, it's actually whether we could play with biological processes such as mitophagy. So mitophagy, it's a process that leads to degradation of damaged mitochondria. And this process is actually triggered by the accumulation of a protein called PINK1, in damaged mitochondria upon damage of the membrane. And upon accumulation of PINK1, you have actually a recruitment of a protein called parkin, then will lead to the formation of autophagosome and ultimately to the degradation of mitochondria by the lysosomes. And so we were wondering whether simply by accumulating PINK1 at the surface of healthy mitochondria, we could actually induce mitophagy. So what we did, it's very simple, as we can really just use catch fire to recruit protein at the surface of mitochondria. With this time just fused PINK1 to the fire tag. We also included M-cherry fluorescent protein just to be able to monitor the localization of the protein. And so in absence of match, what you see, it's very healthy mitochondria. However, when you add match and you image 2 hours after incubation with a match 550, you see accumulation of PINK1 at the surface of mitochondria, and you see a clustering of mitochondria, which are indicative of induced mitophagy. And we could actually quantify the induction of mitophagy simply by characterizing the area of the mitochondrial network. And we see really a decrease of this mitochondrial, the area of this mitochondrial network, indicative of induced mitophagy. So this is a very simple experiment demonstrating how we can use catch fire to control a biological process. So then we wanted to go further and see that we could also control protein transport. And we first decided to demonstrate that we could use catch fire to control nucleocytoplasmic protein transport. So as you all know, this nucleocytoplasmic protein transport is controlled by specific sequences inside protein called nuclear localization signals and nuclear export signals that interact with the import and export machinery in the cell. And so we developed a strategy to both control the import of protein inside the nucleus or to control the export of protein from the nucleus to the cytosol. So in the interest of time, I will just show you one example where we control the nuclear import. And to do this, we actually first fuse so a cytosolic protein, M-cherry, to both a nuclear export signal and to a nuclear localization signal and to a fire tag. And when you have the two signals together, NES and NLS, actually the protein is mainly localized in the cytosol. The reason it's only because the nuclear export signal is stronger than the nuclear localization signals. But the interest of having this nuclear localization signal is that anyway, transiently, the proteins still go to the nucleus. It's just that the export is faster and stronger than the import, and at steady state, you have mainly the protein in the cytosol. You still have proteins going in the nucleus from time to time. And now if you co-expressed a nuclear protein like H2B fused to fire tag, you can actually promote the translocation of the cytosolic protein to the nucleus simply by addition of match. Because through a kind of sync effect, you will just have a capture of the transiently the protein that comes to the nucleus is captured in the nucleus, and through a kind of sync effect, you will just induce and promote translocation from the cytosol to the nucleus, as you could see here on this movie, where part of the cytosolic protein arrived in the nucleus. So this is one example of how we can control protein transport between two different cell compartments. So next, we got interested with controlling secretory protein trafficking in collaboration with Frank Perez, who is, as you know, an expert in secretory protein trafficking. And so I think I don't really have to explain you how secretory protein trafficking works, but that protein that are secreted are expressed in the ER, then they go to the Golgi, and then they reach the plasma membrane through a transport vesicle. Of course, if you want to really evaluate and control or have an idea of the kinetics of the different steps, here it's quite challenging because if you just express a protein at steady state, you will have just fluorescence in all compartments because you have an asynchronous process where it's a continuum of process which is a continuum. If you want to be able to really visualize the different steps of this process, you need to synchronize the traffic. And so a way to synchronize the traffic, it's actually to sequester the protein in the ER, and then to be able to release all at once all the protein from the ER to the Golgi and then to the cell surface. So a few years ago, Frank actually developed a technology called Rush that allowed to do this, that allowed to synchronize protein trafficking by sequestering proteins in the ER. And we thought that we could actually adapt this concept of Rush with the catch-fire technology. So here the idea is to have a protein, a reporter protein that you want to control the secretion. You fuse it to fire tag, and you co-express it with a fire mate fused to ER retention signal like this KDEL signal. You co-express the two proteins in presence of the match molecule, so the protein gets retained in the ER, as you can see here with this localization. And then you can use the fact that upon washing the match molecule, you will dissociate fire mate and fire tag to start the proper traffic of the reporter protein. And here is what you see after washing the match molecule. You see the protein in the Golgi, and then later you see the protein reaching the surface of cells. And so here the movie showing this, so here you are in presence of match, the protein gets sequestered in the ER, and upon washing the match molecule, what you see the protein going to the Golgi, and then protein reaching the cell surface. So this really show you also how we can use catch fire to control this kind of biological process. So we could also show that actually we can even control the retrograde transport from the Golgi to the ER if we want. And yeah, I will skip this just because of interest of time. And the last example I wanted to show you, it's actually how we can use catch fire to also control lysosome positioning and or organelle positioning. So not only to transport proteins with catch fire, but to transport organelles. And lysosome was a very interesting organelle for us. So you know that it's an organelle involved in degradative function inside cells. And the localization of lysosome is controlled by interaction with dynein and kinesin. And in resting cells, lysosomes are in general in the perinuclear region of cells. And this localization is due to really the interaction with specific dyneins. And we thought that we could actually control lysosome positioning by actually controlling the interaction between lysosome and a kinesin that would go from the minus end to the plus end of microtubules. And to do this, we express fire tag at the surface of lysosome and fire mate to the KIF17, which is a kinesin family member 17 from neurons. And so in absence of match, you see the perinuclear localization of the lysosome. If now you add a match, you will induce the interaction between the lysosome and kinesin. And what we observe is the anterograde transport of the lysosome to the cell periphery. And if you wash away the match molecule, you can actually see the retrograde transport to the perinuclear region. So here it really also show you how we can use catch fire to really control in time the positioning of organelles inside cells. Okay, so with this, I will skip this part because I think I'm a bit late. And so I just want to show you that we could develop this technology catch fire that allow us to control a different kind of cell activities, control protein localization, control protein transport, protein secretion, organelle positioning. And we also have a new story that will be submitted soon, where we also control other type of biological processes, including gene expression, gene recombination, and different kind of process. And with this, I want to thank my team and my collaborators, so in particular Frank Perez, with whom I developed this technology, and this agency for funding. And I thank you very much for your attention. Thanks again for the invitation, and I'm ready to take any questions.